heartily to a session on basics of PCR by our subject expert, Dr. M. Shankari. She is trained in molecular pathology, and she will be presenting, and she has presented at several national and state conferences. Over to Dr. Shankari for an enriching talk.
Thank you, ma'am. Let me share my screen now. So, good evening, everyone. Today is going to be a simple topic. We'll be talking about the basics of PCR. How is it done? What is the basic technique used? And what are the components of PCR? So, with this, there will be a short introduction about it. What are the components? What is the principle, programming, and the basic instrumentation and applications. So, the PCR is polymerase chain reaction. It is a laboratory nucleic acid amplification technique used to denature, renature short segments of DNA using the enzyme DNA polymerase 1, which is an isolate of thermos active. So, over ages, PCR has developed many techniques, and with the progression, like initially in 1953, double-stranded DNA helix was discovered, and the PCR technique was first established in 1985. Whereas Carrie Mullis was the first who introduced this PCR technique and who was later awarded the Nobel Prize. So this thermos aqueous DNA polymerase, which is developed from that, is very much known to be resistant to temperatures, different temperatures. That is the reason this is used in the PCR technique. So PCR relatively focuses on the minuscule segment of this DNA. So first step is the nucleic acid isolation. So the nucleic acid extraction or the isolation can be from various sample types. They can be blood, plasma, serum, pus, body fluids, urine, tissues, any type. So next, the Taq polymerase, which we have spoke about, is preferred because it is heat stable and properties enable it enables the DNA synthesis even with the exposure of the primers. So in this nucleic acid extraction step, initially the DNA extraction was based on the DNA gradient density gradient centrifugation. So which was a labor intensive process and time consuming, but it yielded very low quantity of the nucleic acid. Nowadays, the extraction of nucleic acid is either column based, solution based, silica based. With the emergence of this automation and commercial kits, the process of extraction has become simplified, which eventually led to increase in the yield. So the initial step in the nucleic acid isolation is the lysis, which is where the nucleic acid and the cell are broken to release the DNA fragment. Second is a precipitation. It helps in the separation of DNA from the cellular debris. So in this, isopropyl alcohol or ethanol are added. The negative charges present on the DNA molecule are neutralized by the sodium ions, which render them more soluble. By the addition of this alcohol, it causes the DNA precipitation out. So next step is the purification. So in the purification process, the DNA is rinsed with alcohol to remove the extra debris and the remaining unwanted materials. So when there is without sufficient extraction and purification, the contaminants will be present in the DNA, which will hinder our PCR process. So these are the challenges will be like there will be poor reproducibility and sensitivity. Now, once the DNA extraction is done, we have to check the quality and quantity of the nucleic acid which we are getting. So the quantification of nucleic acid is a very plays a very significant step, as it knows the amount of DNA present, which is essential for performing the PCR. So it involves the measurement of absorption to transmission of light. So the methods are UV spectrophotometry, fluorescence, electrophoresis, and high pressure liquid chromatography. So in this, the different to different degrees, molecules absorb different wavelengths, and the molecule passes through a particular wavelength in which they absorb the maximum. So we have two types of ratios. One is 260 by 280, which is used as an indicator for the purity of DNA. Now, the fluorescence, in case of fluorescence dyes are used. In agarose gel electrophoresis, ethidium bromide is used. Now, the updated technology, the capillary electrophoresis method, it is similar to agarose gel electrophoresis, but it is a smaller and automated version. The ratio of absorbance is usually maintained between 1.8 to 2. Anything more than that indicates contaminants are present in the nucleic acid. So coming to the basic steps of PCR, we have now, as extraction is done, there is a template DNA, which is a double-stranded DNA. So the PCR procedure begins by collecting these small DNA fragments in the tubes. So the first step in it is denaturation phase, where the DNA is heated to 95 degrees centigrade. Now this at this temperature, the two strands are separated. It at this temperature dissociates the hydrogen bonds between the complementary bases. So once this DNA strands are separated, next is the annealing phase, where there is rapid cooling from 95 degrees to 55 to 72 degrees centigrade. This annealing involves cooling of the denatured DNA, where allowing the hydrogen bonds to reform. At this juncture, primers are added and they are attached at the 3' prime. And we have 3' and 5' prime end. So primers attached to the 3'. Then comes the elongation and amplification. So once this primer is attached, then the action of DNA polymerase begins. Elongation happens. So this specified temperatures which are maintained is determined based on the physical and chemical properties of the primers which are used. So what are these primers basically? They are 20 to 25 nucleotides in length, and these, as I told you, annealing allows the primers to bind. So an optimal, once the optimal temperature is reached, where the enzyme induced DNA replication happens, it ensures the DNA polymerase activity properly. So this DNA polymerases synthesizes the new DNA strand from 5' to 3' prime. So producing the stands similar to the template stand, and then the amplification happens. So these are the components of, as I mentioned, these are the components of any PCR reaction: buffer, DNA template, primers, nucleic acids, magnesium ions, and DNA polymerase. So buffer stabilizes the polymerase and DNA and the nucleotides. Primers, we have already looked into it. So there are forward primers and reverse primers. Nucleotides are added to each of the growing chain. So the magnesium ion is an essential cofactor of DNA polymerase, which stabilizes the DNA double helix. So DNA polymerase, as I told you, they act from 5' to 3' end. So PCR amplification with these many uh, components in it is a highly error prone and contamination prone. So to ensure the PCR runs properly, we have controls for better accuracy and reliability. So these controls validate the performance of the test. Now, there are different types of controls which are used in each and every PCR. One is a blank or no template control, where there are all the reagents but no DNA template. Negative control, positive control, and internal control. So this internal control, it is to co-amplify the genomic region using the same temperatures, reaction conditions, and the reagents, except that there are no primers. This is an independent control. So as you can see in the picture, This is the programming of the PCR, where initially the temperatures are very high, then drop down to 55 to 72 degrees, then again increases for each and every cycle. So in a PCR graph, we have three stages. One is a geometric or a geometric phase or exponential phase. Next is the linear phase, which declines from cycle to cycle. Then the plateau phase, where there is where there is very less appreciable target amplification. Ideally, this is a sigmoid shaped graph. Now, as I told you, because of so many components in the PCR inhibition happens because of three main things. One is the reverse transcription inhibition, solubility inhibition, or tag DNA polymerase inhibition. Whenever there is an excess DNA input, it also inhibits the PCR. Or when there are some complex structures, or when our target itself is very long targets, it inhibits the PCR. Then the primers. When there is primer dimer formations or excess DNA polymerase, as you know, even the excess magnesium concentration, magnesium is a cofactor which acts along with the DNA polymerase, it inhibits the PCR. So nowadays we have this hot star DNA polymerases which enhances the specificity. They have no activity at room temperature, but only when the temperature increases, they act. So the thermal cycling conditions are very important for the PCR setups. As you have seen, in each and every step is temperature dependent. Now, there can be some sort of false positive amplifications, which can be due to crossover contamination or carryover contaminations, which can be by use of pipette tips without barriers, etc. Then there is the cycle threshold. Cycle threshold is an intersection between the amplification curve and the threshold. It gives a relative measure of the concentration, and it increases with the decreasing amount of template. So coming to the instrumentation part of PCR. So the parts of PCR, basically there is a thermal cycler which has a thermal block. Tubes holding reaction mixtures are there. Then comes the heated lid, which prevents the condensation of water from the reaction mixtures. So we have the reaction module, optical detection system, and the instrument software. In the reaction module, the samples are heated and cooled to precise temperatures, as we have already seen. And each round of amplification, the temperature rises and then cools. In the optical detection system, there's it uses the fluorescence to monitor the DNA amplification in real time. So now this coming to the reaction module, as I've told you, there is a Peltier or the cooling units are there, which rapidly and precisely change the temperatures of the thermal block according to the PCR protocol. So depending on the system, it can be Peltier based or air chamber based, which is used in case of Rotagene. Now, heating and cooling pads maintain the temperature, uses tubes which are suspended. Then we have this optical detection system, where there we have light source, filters, detectors, and optical path. So the light source, it provides the excitation light needed to make the fluorescent dye emit the light. So there are filters, as you can see. It isolates a specific wavelength of light. Now the light source can be LED, halogen, or anything. And the filters, they isolate the specific wavelength that excites the fluorescence. Then comes the detectors. Detectors, what they do? They measure the intensity of the emitted fluorescence. It could be photodiodes, photomultiplier tubes, or CCDs. Then, yeah, this is about the detectors. They capture the image of the reaction flare. Okay. So this optical system, it can be single channel, multi-channel, as nowadays, or digital PCRs also. So there's an instrument software where there's a protocol chapter which you have seen initially. Then the plate setup, data collection is there. The data analysis as I've shown you in terms of graphs. So the applications of PCR, it can be RNA-based applications or DNA-based applications. Gene expression profiling, allelic discrimination, copy number variation, methylation, for simple viral quantification or any sort of detection also, these are used. So various types, different types of PCRs are available. That is allele-specific PCRs, conventional PCRs, digital PCRs, fast cycling PCRs, inverse PCR, methylation specific PCR, nested real-time PCR, reverse transcriptase PCR, etc. So advantages are, see, the digital traditional PCR has a poor precision because it is in agarose gel detection, where it is size-based discrimination happens, and it is only qualitative method. No quantitation can be done, low sensitivity, and it has low resolution. Then comes the real-time PCR. The dynamic range of detection it has improved a lot, but it collects the data in the exponential growth phase of PCR. The values are or the concentration of, for example, in viral cases, they are calculated based on our targets. So the increase in the fluorescence signal is directly proportional to the number of amplicons which are derived. So when coming to digital PCR, the precision is very high. Improved precision is there, and absolute quantification is there. The percentage of wild to mutant in case of any mutations is given clearly here. The fractional abundance is seen in case of digital PCR. Thank you.
Thank you, Dr. Shankari, for your excellent talk. You covered the topic very, very well. Now the session is open for questions.
Thank you. Speaker.
Don't seem like we have any questions.
Yes.
Okay, thank you.
Thank you so much.
Thank you.
Thank you.